So in plants, we have Plasmodesmata. Why? One moment, please. All right, so Plasmodesmata are going to be in plants, and the pens are not working, making my life. The gap junctions are going to be tunnels between the animal cells. Things that are important about the Plasmodesmata and the gap junctions is they will allow very quick exchange between the cells, and particularly gap junctions, we'll see this in two areas. When we have neurons that are directly connected, and so this would be what's known as an electrical synapse. So they don't use the neurotransmitters to cross, but then we also see this in embryology. And then, so the cells that produce the chemicals that control development pass through these gap junctions. So those chemicals are called morphogens. In plants, they'll directly transfer nutrients from cell to cell, but this will also directly transfer the immune chemicals. To fight off viruses, and also to help make the plants taste bad to herbivores and things like that. And so this is just very quick communication between cells. We also have surface-to-surface contact where the cells are binding to each other. The main place where we see this is in the immune response with the major histocompatibility complexes 1 and 2. And so that's going to be cell recognition by the T cells looking for cells that are infected, and also B cells and their antibodies. And so if they connect with a cell and the signal is not correct, then the immune cell knows we need to destroy this one. But we'll also see how this plays out in the fertilization process and how we have that connect. Yes?
What does MHC stand for?
Major histocompatibility complex. So those are the proteins that display antigen components. So an infected cell displays viral particles saying, "I'm infected, kill me." A macrophage also displays those particles to activate the helper T cells, to activate the B cells and the T cells. All right, so distant contact, we have local distant contact. And so we have that in our chemical synapse. So when our action potential gets to the end of our presynaptic cell, causes the release of the neurotransmitters, which activates our postsynaptic cell. We also see this in different cells that secrete chemicals right around them. This could be hormonal, or this could be what's happening right here, right now with my sinuses. We've got, this is when your body is releasing histamines as a response to allergens. But this is also an immune response going on as well, because yesterday when I opened up my composter to dump in my food scraps. I got hit in the face with a big burst of mold spores. And so now I think I have a sinus infection. So that's why I sound like this. But that is because those mold spores triggered a response, and then those cells are now releasing inflammatory chemicals, which are making everything just rather gross. All right. Exactly.
Yeah.
So our hormonal system, this is when we have a longer distant communication. We have some type of endocrine cell that releases a hormone into the blood. The blood transfers that until that hormone finds its target cell. And when we say the target cell, that is the cell where the hormone is going to bind, whether on the surface or interior, and it's going to initiate the signal transduction pathway. And again, we'll be talking about this, but that's where the hormone binds, that's the reception. Then it causes some kind of interior cellular change, which is the transduction pathway. And then that transduction pathway is going to initiate a cell response to that hormone or to that signal. So when we talk about hormones, again, because this shows up all the time on the AP test, if it is a protein or an amine hormone, it is binding to a surface protein. Because this hormone cannot cross the cell membrane. If it is a lipid hormone, that is going to go through the cell membrane, even through possibly the nuclear membrane. Because it is a lipid hormone, it can pass through the cell membrane, and it has an interior receptor. Whereas the receptor for the protein amine hormone is going to be on the surface. So amine hormones are just a few amino acids, or maybe even one amino acid, where protein hormones are just a little bit longer. All right. Same thing with steroid hormones, which are lipid hormones. They go through. Okay, your general signal transduction pathway, we've got reception. So this is where we have our signal molecule. This is going to be known as your ligand. And this is your ligand receptor. So each ligand has to have its own receptor. Once this happens, this is going to then activate a molecule, which will activate a molecule, which will activate a molecule. That's the transduction. It's just passing the baton of like sending the signal down through a series of proteins. And then the final molecule will then cause some type of cell response. So you can have lots of different things that can go wrong in this pathway that can lead to misinformation to the cell. If we look at, let's jump to here, we have our ligand right here. Our ligand binding domain is that protein where it's supposed to connect. If this is, some mutation causes this to be incorrectly formed, and it's got spikes or whatever, it's no longer going to fit in here, and then you won't get the initiation of the signal transduction pathway. Another problem could happen is this could have the wrong shape, and then that won't fit in there, and you won't get your signal transduction pathway. Other things that could happen, once this binds, it's supposed to be broken down. If this stays attached, or if this protein stays around for a longer period of time, then you could get a higher rate of signal transduction or if you have a cell that's making way too much of the ligand, and if that ligand initiates cell growth, then these are just different ways that cancer could arise. If we look at this picture, which you don't have in there, two things that I want you to see is you can have the same hormone, so epinephrine, that's adrenaline, but you can have different receptors. And then when you have different receptors, that will cause different responses. So here, in your intestinal blood vessels, this causes constriction of the blood vessels, so that takes the blood away from your intestines. And then your skeletal muscle blood vessels dilate, bring more blood to your muscles. So this is why, so then that's going to put more blood, oxygen, and nutrients go to your muscles so you can fight or fly. And then this is going to pull it from your digestion because you don't want to put the blood there. This is also why when you get nervous, which is a small fight or flight response, you get queasy because the blood is pulling away from your stomach. But what we see over here, we've got the same hormone again, but the same receptor, but different types of cells, and you'll get a different response. Because what's different here is the transduction pathway and the ability of the cells, because they are different in how they respond. So here you're going to get glycogen breakdown to make glucose to give you energy. But so you can have multiple different things happening with the same hormone, same responses, or same receptors. All right, transduction, basically what is happening in transduction, we're having lots of conformational changes. And so one protein gets changed, which causes another protein to change, which causes another protein to change. And there's two main things that are going to happen. Or your main mechanisms. And so your main mechanisms are going to be phosphorylation and signal amplification. And so the way that the proteins get activated through the cell is going to be by phosphorylating. And when you phosphorylate, phosphorylation causes a conformational change in the shape of the protein so that it will then activate another protein. But as this is going on, so if you are doing, let's just say 5 steps, and you have one molecule here, this one molecule will phosphorylate 10 molecules, which will then each phosphorylate 10 more, which then might phosphorylate 10 more. And so you get, and it could be even a greater amplification than this. It could even be more extreme. And so one molecule could potentially activate lots and lots and lots, and that's what we mean by signal amplification. So you don't need to continuously, to get a large enough response from your cell, you only need one signal to do that. And so that's what's known as a phosphorylation cascade. So this signal molecule activates your receptor, which activates this, which activates these, and so we're passing these phosphates down and down. And what I want you to, again, kinases, kinase, phosphofructokinase, that equals phosphorylation. So, and then as we are going down this phosphorylation cascade, that will amplify the signal. And so what we see in this one, we see the number of molecules being activated increases. And so you've got lots of things amplified. 2 things you need to be aware of that show up on the AP test, typically as such and such and such is a G protein kinase mechanism, which just kind of like, what does that mean? All it means is it's a signal transduction mechanism. You won't have to explain what and how a G protein kinase mechanism works. Just be aware that it's a signal transduction pathway. Same thing with cyclic AMP. Cyclic AMP is when ATP gets converted into this form, and then this is an activator in the transduction pathway. So we can see here when you've got low, so this is what happens in response to the adrenaline and how the glycogen gets broken down into glucose. It activates the cyclic AMP response, which then allows the fight or flight. So just be aware that they are both parts of the signal transduction pathway. Basic cell responses, secretion of chemicals. It could be proteins, it could be enzymes, it could be antibodies, it could be mucus. This could initiate the building of specific enzymes or proteins. It could be activation of cell death or apoptosis. It could initiate growth. It could change the cell structure. This is one of the ways that your white blood cells can navigate through your body. They get a cell signal and they move out of your blood vessels and migrate through your body tissues. And so all of these different things happening, a lot of it has to do with activating transcription factors. And some of those lipid hormones that pass directly through the cell membrane, through the nuclear membrane, will bind directly to the DNA and act as a transcription factor to initiate some specific response. So these are the main things that will happen. As all this process is going on, you need the signal, you need the transduction mechanism, you need the recepting mechanism, and all of those things, all of them involve proteins. All those proteins have to be made from DNA, RNA, etc. And so you can get all of these different possible things that can mess up your signal transduction pathway. One thing is you can have a new promoter that makes a lot more of your signal. And if you get a lot more of your signal, that is going to cause you to have more and more responses. Or you can have gene amplification. So it's not that one gene is making more, it's just you've got more genes. So this would be an error in crossover where you get an extra gene. And so then you are, again, making more of that signal. Or you get some point mutation farther upstream, which controls transcription. And then the transcription is happening faster and more often, or you get a hyperactive or long-lasting protein due to some mutation. All of these things increase growth factors, increase cell stimulation, which then causes the cell cycle to go out of balance. And if your cell cycle goes out of balance, that's going to be a potential mechanism to stimulate cancer. So whether it is a mutation causing all of these things or some kind of regulator right there, either way they are making too much of your signals. So how does signal transduction factor into fertilization? Your reception, the reception part is right here when the sperm proteins bind to the surface of the egg. Of course, that has to match, so that's how we keep species separate. This is a prezygotic barrier, so if the sperm can't fuse with the egg, then you're not going to mix. And then what happens when that fuses properly, you can see right here, the membrane becomes depolarized. This is going to be the transduction part. And this is the reception part here. So what this depolarization does, that changes the charge of the egg membrane, and then the response of that is the cortical reaction. And that's where all of these granules spread in between the fertilization envelope, and then water flows into that salt, separating the fertilization membrane from the cell membrane, and that prevents two sperm or multi-sperm from fertilizing one egg.
Can you say that again?
Yeah, can you say that?
The salt part?
So the cortical reaction is that these cortical granules release salt between the fertilization envelope and the cell membrane, and then our rule, water follows salt. It's actually calcium ions, but calcium ions are salt. And then that causes water to flow in and separate them.
Separates the embryo.
What?
What is it separating?
So you've got this fertilization membrane with your receptor proteins, and then you've got your cell membrane underneath. And so when it separates, it breaks the receptor proteins away from, so then sperm, if these receptor proteins aren't attached, then your sperm can't fuse with the cell membrane of the egg cell. All right, so, excuse me. A lot of this can lead to positive and negative feedback loops. So a negative feedback loop. So this is a counter response. So this is when you reach a certain level and then the opposite thing happens to kind of slow that down. So it's not always that it's lowering something. Sometimes the counter response is raising something. So with blood sugar levels, we have two types of negative feedback. So we see that when your blood sugar is too low, and that's when it drops below 90 milligrams per 100 milliliters or 1 deciliter, then your pancreas secretes the glucagon, which breaks down glycogen. That releases your glucose into the blood, raising it back up. If you go to lunch and you have a big bowl of pasta, as your body digests the pasta into glucose, your blood glucose will get too high, which is above 90 milligrams per deciliter. And then your body is going to release insulin, and that's going to cause your body cells to uptake that glucose and store it until it's back to that 90 milligrams per deciliter. Both of these are negative feedback mechanisms. Even though one is going down and one is going up, they're both negative because they are countering the opposite thing. Body temperature, when you get too hot, you're going to sweat. When you get too cold, you're going to shiver. Both of those are going to be, again, negative feedback because you're countering what's going on. All of this is regulated by your hypothalamus. That's your internal thermostat. And it will also affect your blood vessels as well. When you're too warm, you vasodilate, getting that blood to the surface of your face and your skin. Or you vasoconstrict when you're cold to pull that blood away from your skin so you don't lose as much heat. In your thyroid, again, going to our thyroid stimulating hormone. Your thyroid stimulating hormone is going to tell your body to make T3 and T4, triiodothyronine and thyroxine. For these, you need your iodine. So normally when you have low T3, T4, thyroid stimulating hormone tells you to make these. and then you've got your normal levels. If you don't have enough iodine, then that keeps activating your thyroid, and what does that lead to? That leads to the goiter. What?
There's someone with a goiter in our book.
Yeah.
There it is.
All right, some positive feedback. So birthing, labor. So this begins with the release of oxytocin. This stimulates the uterus to contract. And then that stimulates the release of oxytocin. So a positive feedback isn't a countermeasure, it's an amplifying measure. What happens if you hold the microphone in front of the speaker? You get the feedback because the sound is going through and feeding back on itself and amplifying out. And so the same thing. And so then what happens is then you keep doing this and doing this and doing this. More oxytocin, stronger contractions, more oxytocin, stronger contractions. Baby. All right. In the environment, we also have some positive feedback mechanisms that may not actually be good. So as surface temperature, we get some CO2, we get increased surface temperatures, and so then up in the Arctic, your permafrost becomes not permafrost. So permafrost means permanently frozen. That permanently frozen soil traps methane and CO2. As that permafrost starts to thaw out, then it releases that trapped carbon in the form of CO2 and methane. Both of those greenhouse gases, methane way, 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 way more potent than CO2. And then you get more warming, more melting, more release, and so then it is a negative. As that warming increases, what's going to happen is your polar ice will melt as well. Polar ice is important because it is very white and very bright, and it reflects sunlight. That is known as the albedo effect. And so as the albedo effect lessens, more energy is absorbed instead of reflected. That causes stuff to warm, which then increases this whole process. So sometimes you will see in new construction on larger buildings, they will actually paint the roof white. And so that will reflect light. That helps with cooling costs because then that is not absorbing heat energy. Functions of mitosis: growth, repair, and asexual reproduction. So you were once one cell, now you're about 37 trillion. So we had to do a lot of this mitosis stuff for you to be here. And so then when we talk about sexual reproduction, that's where we'll get into meiosis. But your main function or your main phases of mitosis, you've got interphase. G1 is simply where you're going to have the growth of the cell. S phase is where we're going to synthesize our DNA replication. And G2 is basically our DNA check and our prep for division. And so as we are going through interphase, there are going to be multiple checkpoints and stages. Sometimes you will have a cell that will go into G0 stage. G0 can be one of two types of cell rest. The two types of cell rest are going to be the quiescent phase. This is basically, we're just taking a break. We don't need to actively divide all the time. So your neurons, they're not going to divide anymore. They are in their quiescent stage. The egg cell, after that first burst of development, they stay quiescent until they are stimulated by the follicle stimulating hormone. When cells go into senescence, they're done. You can exit quiescence and go back to dividing. Neurons don't do that though. But senescence is when your telomeres have gotten too short, the cells can't divide anymore, and so they are going to be essentially turned off in terms of division. Our basic phases, what we need to know in prophase, our most important thing is that our chromosomes condense so that they can be separated. Yes, the nucleus disappears, that's important. Yes, the spindle fibers start to grow, but your most important thing is the condensation of the chromosomes. Prometaphase, this is where your spindle fibers attach to your centromere. That's because we need that to happen so that they can be separated. Metaphase, they're lining up in the middle. This is where we have our spindle check. That is actually what is most important, to make sure that each centromere, each kinetochore has a spindle fiber attached. And then an anaphase is where the centromeres the chromatids separate.
Sir, we have 2 days for this, right?
No, don't worry. There's a video online as well. So, and then in telophase is where our nucleus reappears. So, unlikely that they'll give you a free response, say, explain all the steps of mitosis. They're not going to do that. But you may have to be able to identify those phases, like you did on your test when we had the cell division test. So when you do a little drawing of each of the basic, what each stage looks like as part of this. So we then get into cytokinesis. So animal cell cleavage furrow, just pinches in half. Plant cell, we have a cell plate, which then develops into the new cell wall, and that grows and divides them in half. Our major cell checkpoints, we've got the G1 checkpoint. This is basically based on CDKs, cyclin-dependent kinases. Which we'll talk about in a second. We have the G2 checkpoint. This is where we're proofreading the DNA. And then metaphase is the spindle attachment. So these are what regulate the cell to make sure everything should be happening in the correct order, and then the correct timing to make sure that the cells have grown enough. That's what the cyclin-dependent kinase checkpoint does. Is the cell big enough to divide? This checks to make sure the DNA is correctly copied, and this checks to make sure that the spindles will actually separate the sister chromatids so you don't get non-disjunction. What's non-disjunction? Right. So instead of separating, then they kind of get stuck together and go to one side. So cyclin-dependent kinases and cyclin. So our cyclin-dependent kinases, they are always present. And then our cyclins, they build up. And so when you get a high enough concentration of cyclin, they bind to your cyclin-dependent kinases, and that activates the next cell cycle. So that's going to promote division. And so that takes time for that cyclin to build up. And so that gives the proper amount of time for the cell to get big enough so it doesn't keep dividing, and the cells don't get too small. So that's what all these, so different types of cyclin-dependent kinases. Things that can happen incorrectly, you can have errors in replication. That's going to activate this P53 protein. What you need to know about P53, this is activated with cell damage. And then it stops the cell cycle. So it's either going to initiate DNA repair or what? Or apoptosis.
That happens a lot, right?
What?
Does that happen a lot?
It does. Yeah. So you're losing lots of cells to this all the time, especially if you get sunburn. Like that's the whole inflammation and the peeling process is because of that. All right. So rolling through our hormones real fast. Antidiuretic hormone is going to be for water retention. When your blood is too salty. So you've sweated a bunch, you haven't been drinking enough, so ADH gets released, and that targets the collecting duct. And so that causes the collecting duct to absorb more water, so then your pee is going to get darker. Calcium regulation is going to be controlled by calcitonin and parathyroid. So calcitonin is going to lower blood calcium, and this is going to increase blood calcium. And your general level is 10 milligrams per deciliter. So if you drop below that, parathyroid gets released to break down your bones. If you are above that, calcitonin, remember calcitonin tones your bones and makes them stronger. It absorbs the calcium into your bones. It also plays into vitamin D activation and the role of the kidney absorbing and secreting calcium. Insulin and glucose, when you are above, we already talked about this, then your body breaks down, or when your glucose is above, your body absorbs it with insulin. When it's below, glucagon causes the breakdown of glycogen to release the glucose into your bloodstream. Melatonin is for regulating your circadian rhythms. This is both genetic and environmental. So the light stimulates the blocking of melatonin production from the pineal gland. Darkness stimulates the release of melatonin, and so it's the light-dark cycles that regulate your circadian rhythm. If you remove that external, then you'll have a natural genetic circadian rhythm, but they are not the same. So the external environment actually can override your genetic levels. So quickly, luteinizing hormone, Leydig cells, luteinizing, Leydig, that makes your testosterone, that promotes spermatogenesis. 1 minute. We'll do the whole ovarian cycle. Follicle stimulating hormone, Sertoli cells, that develops the sperm. So, stimulating hormone, S, sperm, Sertoli, all of that together. So this is just a constant process in the males. When we get to the female menstrual ovulation cycle, you're going to have to come back to this, but your follicle stimulating hormone matures your eggs and releases the estrogen. So that's going to then target the development of the endometrium of the uterus. And the release of the luteinizing hormone. That luteinizing hormone causes ovulation, and then the corpus luteum, which makes the progesterone to maintain the endometrium during pregnancy. No pregnancy, corpus luteum breaks down, uterine lining breaks down, causing menstruation. Other things, adrenaline, this is for your fight or flight. We've talked about that a lot, releasing, causing all these different responses. Glucocorticoids are also a response to long-term stress. And cortisol will then eventually suppress your immune system. Plants, things to know, go through stem growth and root growth for auxins, opposite for cytokinins. Gibberellins, the important thing is that is for seed germination. Ethylene is basically for the breakdown of tissues. And abscisic acid is for dormancy and protection. So for a seed to germinate, you need to diffuse the abscisic acid out. That's what the water does. Then the gibberellin gets activated and it promotes the seed to germinate. All right, so you're going to have to roll through the end of that bit on your own. This video, at some point, we will watch. It's a great video. You might want to watch it. It's about how bacteria communicate and glow in the dark. It's super cool. So.
These are not